So in the case of liver, we have to study about two aspects. One is structure or anatomy of the liver and histology. It means what are the different tissues present in a liver. So here you can see that some basic features about the liver. It consists of largest internal organ in the body. That is one aspect. Then the weight of the liver is coming to 1.5 kilogram. Now, embryologically, during the time of embryology, you might have studied that there are three regions: foregut, midgut, and hindgut. So here, the development of a liver actually takes from foregut region of the embryo, and it spans on the upper right side, most of the time just above the stomach, and part of the left abdominal quadrant. So this is regarding the position of the liver. Now if you take a liver, it consists of four lobes, two larger one and two smaller one. Maybe so this is the nature of the liver. So these are all lobes. There are four lobes are there, two smaller lobes and two larger lobes. Right and left lobes are known by the name larger liver lobes, and quadrate lobe and caudate lobe. These are the two smaller liver lobes. So this is an idea regarding the basic structure of the liver. Now physiologically speaking, liver, the main function of the liver is actually producing the bile. Bile is an important digestive fluid, and that bile is actually what is helping in digestion of the fat. So liver has therefore an important function, mainly regarding the digestion of the fat. So here you can see it has performing many important function. Then here also you can see how we'll come to the histology part of that. See, histologically each liver consists of hepatic globules separated from one another by a thin layer of connective tissue known by the name Gleason capsule. So this is how the tissue level arrangement of liver. So there are number of lobules are there. So and each liver is actually formed of thousands of lobules. So this is the structure. This is the main structure. And if you cut the liver, you can find and this kind of features, and if you want closer examination, you can see that each liver is having a hepatic globule. So hepatic globule is one of the primary or basic unit of liver structure. Now it consists of a number of hepatic globule, and they are separated from one another by a thin layer of tissue called the Gleason's capsule. Here you can see that. So this is known by the name Gleason's capsule, and this is the hepatic globule. So this is how the liver is actually arranged, and there is a thin layer of connective tissue which we call by the name Gleason capsule or simply septa. So the lobules are just visible to naked eye and are hexagonal in outline. See, so this is the lobule. They have a hexagonal shape. So we have to see what are the important structures present there. So in the center of each lobule lies a hepatic vein, the intralobular vein, otherwise known by the name central vein. Here you can see that. So this is the intralobular vein or central vein. We can also call it by the name hepatic vein. So there is a central vein on the hepatic lobule. Then the cells forming the lobules are cuboidal or polyhedral in shape and are called hepatic cells. So here you can see that if you are taking these cells, each of the small is known by the name hepatic cell. Their shape is actually either cuboidal or polyhedral in shape. So that is they do not have a proper shape. That is why polyhedral is given. So each hepatic globule will be having thousands of liver cell or hepatic cell, and at the center we are finding a central vein or hepatic vein. Then these are arranged in radial rows of one layer or two cell thick rods, or trabeculae, which extend from central vein to periphery of the lobule. See here, you can see that. So this is how the whole arrangement is here. So these are all hepatic cells. This is the central vein. Now what we are seeing is here you can see that they are arranged in radial rows of 1 or 2 cell thickness or trabeculae, which extend from central vein to peripheral lobe. See, so here you can see that this entire portion is known by the name trabeculae. Their thickness may be either 1 cell in thickness or 2 cell in thickness, and that projection from central vein to the margin is known by the name trabeculae. So that is the idea of hepatic lobule. So there is so you can see that histologically it consists of hepatic lobule. Hepatic lobules will be separated from one another by Glissen's capsule. Then there is a central vein. Then number of hepatic cells are there, and what we are seeing is the presence of what we call trabeculae. So presence of trabeculae, which are 1 or 2 cell in thickness. So this is an idea regarding how liver cells are actually arranged. So hepatic cell possess fine granular cytoplasm, large round central nuclei, and deposits of glycogen and lipid. So these are the substances that we can find present inside this structure. Then between the hepatic cords is a labyrinth of narrow irregular spaces called lacunae. See here you can find that. So this space is known by the name lacunae. So you can see that this is the lacunae, and through which run blood capillaries known by the hepatic sinusoid. See what we are seeing here, this is the sinusoid. So between the hepatic cells we can find blood-filled spaces. These blood-filled spaces are known by the name sinusoid. So that is how another important aspect of the liver structure is mentioned. Then the wall of the sinusoids are discontinuous and made up of endothelial cell. Then we have another type of cell known by the name Kupffer cell, and their function is to ingest or eat the dead cells as well as erythrocytes or RBC, and destroy the harmful bacteria that may be entering into the liver. So that is another kind of cell known by the name Kupffer cell. Here you can find that. This is the Kupffer cell. So Kupffer cell will be feeding on other bacteria, and they will be also protected, also feeding on RBC as well as some other kind of cells are also taken by the Kupffer cell. And at the corner between adjacent lobules are strands of interlobular connective tissue called portal areas or portal canals. So here also you can see that there will be portal canal somewhere here. So this is the portal vein, this is the portal canal, that is how. So this is the hepatic vessel or central vessel. Here you get a central vein or hepatic vein, and this one is the intraportal vein. This is what we are seeing here. So in addition to that, portal canals also contain nerves and lymphatic vessel. Then also another feature is that sinusoids containing blood from intralobular branches of hepatic portal vein and hepatic artery, they are emptying into intralobular central veins which unite to form hepatic vein. See here you can see that whatever secretions are secreted by liver cell or Kupffer cell or liver cell, that will be directly pumped into the central region. So it is from the central hepatic vessel the most of the secretion or especially bile is actually taken by the body to various organs of the body, especially digestive system. So this is an idea of how liver cells are actually working. Then coming to the histology part. So here you can find that the liver cell consists of four major type of cell. One is parenchyma, second one is stroma, and third one is a sinusoid, and fourth one is the space of Deci. So here you can see that. So this is how the entire idea is. The one is parenchyma. Parenchyma is, this is the most of the liver cells are actually parenchyma or parenchymatous cell. Then we can also see that stroma, which is actually a continuation of surrounding glissons. Say here you can see that this is the glisson capsule, and that portion is known by the name stroma. So this is another portion. So stroma, which is a continuation of the surrounding capsule of glisson. Then another area is sinusoids, which are capillaries traveling between hepatocyte. Here you can find this red colored portion. That red colored portion is known by the name sinusoid. Sinu means blood. So sinusoid means blood filled cavity. So in the liver there are certain blood filled cavities known by the name sinusoid. They are normally present between the liver cell. Then, so this is an idea how liver cells will be arranged. And finally we have space of Deci or perisinusoidal space, which are located between hepatocytes and sinusoid. So here you can find a space of Deci. I cannot show you here. There are no proper picture to show the space of Deci. So in case of space of Deci, they are located between hepatocytes as well as sinusoid. So here, see here you can see that these are the space between hepatocyte and the sinusoid are known by the name space of Deci. So this is an idea regarding how the various organs, various tissues within the liver are actually arranged. So this is the first type of gland, that is the liver.